Hi everyone, this is Annie and I'm presenting research done at a lab at the Australian National University, published in 2017. This paper details the lab's use of ancestral sequence reconstruction to gain insight into the sequence structure-function relationship for biocatalysts. As a quick review, ASR infers protein sequences that most likely gave rise to the modern protein of interest by using multiple sequence alignment. By using ASR, researchers can see what mutations have occurred in the past and test how these mutations affect function and stability of proteins. So the protein the authors were looking at is the transaminase biocatalyst, and you can see it in the middle. It's used in a variety of reactions in the chemical and pharmaceutical industries. Specifically, it's used for chiral building blocks and improving pharmacological properties of antibiotics. Unfortunately, this biocatalyst is notably inefficient and requires multiple stages of reaction and production lines and has a low activity compared to other enzymes. So right now, many production lines rely on crude oil products to make the process more efficient. But an improved biocatalyst would allow for reduced dependence on crude oil and provide more environmentally friendly solutions and a move towards green chemistry. Protein engineers have tried improving the biocatalyst through various approaches, including random and site-directed mutation, but they haven't had much success. So the authors turned to ancestral sequence reconstruction to see if any insight could be gained. They focused on 192 ancestors and specifically the 6 nodes shown on the subset of their phylogenetic tree on the left. From these 6 nodes, they examined the non-conserved residues to gain insight on how each mutation affected the activity and stability of the biocatalyst. The non-conserved residues are shown in red on the enzyme model. And you'll notice that they are spread all across the enzyme. They aren't localized, which is interesting. So they use this, the reaction on the bottom right for screening. So they tested each ancestor on the modern biocatalyst in an alanine dehydrogenase coupled assay to test activity against 40 different substrates. They found that the modern biocatalyst showed activity for 27 of the substrates, while the ancestor sequences showed activity for 36 of the substrates. This was expected since ancestor sequences are typically more promiscuous than modern sequences. The graph on the right shows the results of the activity assay for the modern biocatalyst and the 6 ancestor sequences evaluated. The spokes for each graph show relative activity for a subset of substrates, and the spoke position is the percentage of relative to the highest activity for that biocatalyst. The researchers found that the ancestral proteins were more active for 80% of the 40 tested substrates, and that the activity was increased by 20-fold for some substrates. Another trend they noticed was that the modern biocatalyst showed preference for omega amino acids, while the ancestor biocatalyst showed preference for alpha-omega diamines. Additionally, the ancestor sequences showed higher thermal stability, measured by differential scanning fluorometry, which is also expected of most ancestral sequences, although it has been shown that it's not necessarily a given truth that ancestor sequences are more thermally stable. After identifying the non-conserved residues, the researchers noticed that a majority were not in the active site. So this is a trend that is uniquely exposed by ASR. A lot of random and directed mutations often focus on the active site and thus limit the mutational sequence space. The authors note that this is a major advantage to using ASR since they can explore more of the sequence of the protein and how that affects the function and stability. The authors also commented that ASR is especially helpful when empirical understanding is lacking, as it can provide guidance for mutations outside of the active site that aren't intuitive to know. These non-conserved residues can be used to gain further insight to the sequence structure function relationship of biocatalysts. Thank you for listening.